There is a creature sitting at the bottom of a shallow tide pool right now, somewhere off the coast of Australia, that weighs less than a golf ball. It is no bigger than the palm of your hand. Its skin is a dull, sandy brown. And if you were wading through that tide pool barefoot, you would step right over it and never know it was there. But inside that tiny, unassuming body is enough venom to kill 26 full-grown adults in a matter of minutes. There is no antivenom. There is no antidote. And the bite, you probably would not even feel it. Before you could register what happened, the toxin would already be threading through your bloodstream, silently shutting down every voluntary muscle in your body, one nerve at a time. Your legs would give out. Your arms would go limp. Your chest would tighten as your diaphragm stopped responding. And here is the part that separates this venom from almost anything else on the planet. You would remain completely, horrifyingly conscious through all of it. Your eyes would be open. Your mind would be alert. You would hear the waves, feel the warmth of the sun on your face, and understand with total clarity that you are suffocating to death while trapped inside your own motionless body. This is the blue-ringed octopus. And that venom, Tetrodotoxin, is estimated to be somewhere between 1,000 and 10,000 times more potent than cyanide, depending on who you ask and how you measure it. An amount that would fit on the head of a pin, roughly 0.5 milligram, is theoretically enough to end a human life. The creature carrying all of this chemical devastation is about the size of a tangerine. Tonight, we are going inside this animal. We are going to trace its family tree back over 500 million years to a time before any dinosaur walked the earth. We are going to look at how a tiny octopus evolved to outsource its most lethal weapon to bacteria living in its own saliva glands. We are going to watch, in detail, how it lights up its body with 60 pulsing neon blue rings in under 1/3 of a second. A warning system so fast and so vivid that it has inspired cutting-edge military camouflage technology. We are going to follow the toxin molecule by molecule as it plugs the sodium channels in your nerve cells like a cork jammed into a bottle. And we are going to sit with a 4-year-old boy on a beach in Queensland who picked one up in a rock pool and nearly died 20 minutes later. The blue-ringed octopus is a masterpiece of evolutionary engineering, and it carries one of nature's most sophisticated chemical weapons inside a body that has no skeleton, no shell, and no spine. It is soft, small, shy, and absolutely lethal. Settle in. This one is going to stay with you. Let us begin with what you are actually looking at when you see a blue-ringed octopus, because it is easy to underestimate this animal based on its size alone. When we talk about the blue-ringed octopus, we are not actually talking about a single species. We are talking about a genus, a family of closely related species, all classified under the scientific name Paralyze. The genus was first formally described in 1929 by British zoologist Guy Coburn-Robson. And as of now, scientists have confirmed 4 distinct species. But that number is changing. Recent genetic research, particularly a major study published in 2023 that analyzed over 10,000 single nucleotide polymorphisms across specimens collected from Australia to Japan, identified a minimum of 11 genetically distinct lineages within the genus. That same study pushed the estimated origin of the genus back to approximately 30 million years ago. So the 4 species we know about today may turn out to be just a fraction of the real diversity hiding in this group. There could be 10 or more blue-ringed octopus species swimming in the Indo-Pacific right now, many of them yet to receive formal scientific names. The 4 we do know are the greater blue-ringed octopus, Hapalochlina lanulata, which is the most widespread, found across Indonesia, the Philippines, Papua New Guinea, Vanuatu, and the Solomon Islands. The southern or lesser blue-ringed octopus, Hapalochlina maculosa, which sticks to the cooler waters along Australia's southern coastline and around Tasmania. The blue-lined octopus, Hapalochlina fasciata, which is found off the eastern Australian coast, and despite its name, does actually have blue rings on its arms, in addition to its signature blue lines along the mantle. And Hapalochlina nearstrassi, a species that has been officially recorded exactly twice in the entire history of science. First in 1938 from a single specimen found in the Bay of Bengal near the Andaman Islands, and again in 2013 when a second individual was hauled up in a trawling net near Chennai in southeastern India. That is it, 2 specimens, 2 sightings, nearly a century apart. We know almost nothing about this species beyond the fact that it exists. In terms of physical size, these are some of the smallest octopuses in the ocean. The greater blue-ringed octopus tops out at about 10 centimeters from mantle tip to arm tip, with an average weight hovering around 80 grams, which is roughly the weight of a small apple. The southern species can reach a bit larger, up to about 22 centimeters, including its arms. When resting, their bodies are a muted palette of sandy yellows, tawny browns, or grayish greens, colors that blend perfectly against the rubble, shell fragments, and algae-covered rocks of their shallow water habitats. You could stare directly at one and not see it. They are that good at camouflage. Like all octopuses, they have 8 arms lined with suckers, a soft and boneless body that can compress and reshape itself to slide through astonishingly small gaps, and a sharp, parrot-like beak hidden at the center of their arm crown. That beak is what delivers the bite. It is small, maybe 2 millimeters across in many specimens. That is about the width of a pencil lead. And yet, that tiny beak connects directly to the posterior salivary glands, which are the production factories for one of the most dangerous biological compounds on the planet. But the most famous feature of this animal is, of course, those rings. Each blue-ringed octopus carries somewhere between 50 and 60 iridescent blue rings across its mantle and arms. When the octopus is calm, those rings are invisible, hidden beneath the surface of its camouflaged skin. When it feels threatened, those rings appear, and they appear fast, flashing into vivid, electric blue in as little as a third of a second. That is not a metaphor. That is a measured, published number. Researchers at Brown University and the Marine Biological Laboratory in Woods Hole, Massachusetts, filmed the display using high-speed cameras and confirmed that full ring expression can be achieved within about 10 frames at 30 frames per second. That kind of speed requires a mechanism that is entirely unique in the animal kingdom. One that does not rely on any chemical signaling at all. And to understand how it works, we need to first understand where this animal came from. Because the story of the blue-ringed octopus is also the story of over 500 million years of cephalopod evolution. The lineage that eventually produced the blue-ringed octopus stretches back to the Cambrian period, over 500 million years ago, when the very first cephalopods appeared in ancient seas that would be utterly unrecognizable to us today. These earliest ancestors were not soft-bodied hunters. They were small, shelled creatures that looked more like snails than anything we would call an octopus. The story of how they became what they are now is one of the most dramatic transformations in the history of animal life on Earth. The oldest fossils that scientists tentatively assign to the cephalopod lineage date back roughly 522 million years to a creature called Tanula, found in early Cambrian rocks. Its status as a true cephalopod is debated, but it had a chambered shell that suggests it was on the road to something more complex. A stronger candidate for the earliest crown group cephalopod is Plectronoceras, found in Upper Cambrian limestone deposits in northeastern China, dating to roughly 500 million years ago. Plectronoceras had a small, cone-shaped shell with internal chambers separated by thin walls called septa. And crucially, it had a siphuncle, a tube running through those chambers that allowed the animal to regulate gas and fluid inside the shell for buoyancy. This was the key innovation. It meant the animal did not just crawl along the seafloor, it could float. And eventually, it could swim. That siphuncle was the cephalopod starter kit, the feature that separated these animals from their bottom-dwelling mollusk relatives and set them on a trajectory toward the open ocean through the Ordovician period, roughly 480 to 444 million years ago. Cephalopods diversified explosively. The nautiloids, with their coiled or straight external shells, became the dominant predators of the Paleozoic seas. Some grew enormous, like the giant orthocone nautiloids, whose straight cone-shaped shells reached lengths of several meters. These were the apex predators of their age, the sharks of the Ordovician, cruising through shallow seas with jet propulsion and seizing prey with muscular tentacles. But shells, for all their protective advantages, came with trade-offs. They were heavy. They were rigid. They limited how fast you could swim and how quickly you could maneuver. And over millions of years, one branch of the cephalopod family began moving in a radically different direction. The colloids, which include the ancestors of all modern squid, cuttlefish, and octopuses, began internalizing their shells, shrinking them down, and eventually discarding them almost entirely. This process took tens of millions of years, but it freed the colloid body plan from the constraints of external armor and opened up entirely new ways of living. Without a shell, you could compress your body. You could squeeze into crevices. You could change shape. And if you could not hide inside a fortress, you needed a different survival strategy altogether: camouflage, intelligence, speed, and venom. The split between the lineage leading to modern squids and cuttlefish, and the one leading to octopuses and vampire squids occurred deep in the Paleozoic. A landmark 2022 study published in Nature Communications described a 328-million-year-old fossil discovered in Montana's Bear Gulch Limestone Formation. A creature named Silipsimopodi bideni, which represents the oldest known vampiropod, the superorder that includes modern octopuses and vampire squids. This specimen had 10 arms, each bearing 2 rows of suckers, and showed evidence of an ink sac, suggesting that defensive ink ejection is a strategy that has persisted for over 300 million years. This fossil pushed the origin of the octopus lineage back by at least 82 million years beyond previous estimates, meaning octopus ancestors were swimming in ancient oceans well before the first dinosaurs ever appeared on land. From there, the octopod body plan continued to refine. The external shell, which had defined cephalopod life for hundreds of millions of years, was reduced to 2 tiny internal rods in modern octopuses. Remnants so small you could easily miss them during dissection. 2 of the original 10 arms were lost, possibly converted into the filament-like appendages still seen in vampire squids, or simply reabsorbed into the body over evolutionary time. The brain expanded dramatically. The nervous system is decentralized, spreading roughly 2/3 of its neurons out into the arms themselves, creating the 9-brained, 3-hearted, blue-blooded body plan we see today. And somewhere along the way, in the warm, shallow waters of the Indo-Pacific, roughly 30 million years ago, a small, soft-bodied octopus lineage began evolving one of the most potent chemical defense systems in the animal kingdom. The genus Hapalochlina was taking shape, and it was about to become very, very dangerous. But before we get to the venom, it is worth pausing to appreciate just how alien the body that carries it really is, because the blue-ringed octopus shares the same fundamental design as every other octopus on the planet. And that design is something that would feel right at home in a science fiction novel. Every octopus, including the blue-ringed species, has 3 hearts. 2 of them, called branchial hearts, sit at the base of the gills and pump deoxygenated blood through the gill tissue, where it picks up oxygen from the surrounding water. The 3rd heart, the systemic heart, takes that freshly oxygenated blood and pushes it out to the rest of the body, delivering oxygen and nutrients to muscles, organs, and the brain. There is a catch, though. The systemic heart actually stops beating when the octopus swims. The muscular contractions involved in jet propulsion interfere with the heart's pumping action, which is one of the reasons octopuses tend to prefer crawling over swimming. Sustained swimming exhausts them in a way that it would not exhaust a fish. You will often see a blue-ringed octopus walking delicately across the seafloor on its arm tips rather than jetting through the water. And now you know why. The blood that runs through those 3 hearts is not red. It is blue. Human blood gets its color from hemoglobin, an iron-based protein that binds and transports oxygen. Octopus blood uses a completely different oxygen-carrying molecule called hemocyanin, which is copper-based rather than iron-based. When hemocyanin binds oxygen, The copper atoms give the blood a distinctive blue color. This is not just a cosmetic difference. Hemocyanin is significantly more efficient than hemoglobin at transporting oxygen in cold, low-oxygen environments, which makes it perfectly suited for life in the ocean. It is a heavier, bulkier molecule, which partly explains why octopuses need those extra hearts to keep the blood circulating at adequate pressure. But in the frigid, oxygen-poor waters of the deep sea and the variable temperature shallows where blue-ringed octopuses live, copper beats iron. The trade-off is that hemocyanin-based blood is extremely sensitive to changes in water acidity. If ocean pH drops too far, which is exactly what ocean acidification driven by rising carbon dioxide levels is causing, the octopus's blood becomes less efficient at carrying oxygen. The animal effectively starts to suffocate in its own body, even if there is plenty of oxygen in the water around it. This is a vulnerability that may become increasingly relevant as climate change accelerates. The nervous system is arguably the most remarkable feature of the octopus body plan, and it is completely unlike anything found in vertebrates. An octopus has roughly 500 million neurons, a number comparable to what you would find in a dog, but the distribution of those neurons is radically different. Only about 180 million neurons sit in the central brain, a donut-shaped structure wrapped around the esophagus between the eyes. The remaining roughly 320 million neurons are distributed throughout the 8 arms, concentrated in clusters called ganglia at the base of each arm, with additional neural networks running all the way down to the suckers. Each sucker on an octopus arm contains approximately 10,000 neurons of its own, giving it the ability to independently taste, touch, and grip objects without any input from the central brain. This is why people sometimes say octopuses have 9 brains, 1 central brain, and 8 arm-based mini-brains. The description is oversimplified, but the functional reality is that each arm can operate with a degree of independence that would be unthinkable in a vertebrate body. An octopus can be exploring a crevice with 2 arms, holding onto a rock with 2 more, eating a crab with another, and keeping watch with the remaining 3, all more or less simultaneously, with the central brain providing high-level coordination, but leaving the details to the local neural hardware. Octopuses are widely regarded as the most intelligent invertebrates on the planet. They can solve mazes, open screw-top jars, remember solutions to puzzles, recognize individual human faces, and display what researchers describe as play behavior, manipulating objects that have no food or survival value purely out of what appears to be curiosity. The blue-ringed octopus, being small and short-lived, has not been studied as extensively for cognition as larger species like the common octopus. But it shares the same neural architecture, and there is every reason to believe it possesses the same fundamental capacity for learning, memory, and problem-solving that has made its larger cousins famous. If you ever get the chance to watch a blue-ringed octopus flash its rings in real time, you will understand immediately why this display has fascinated biologists for decades. The transformation is abrupt and total. One moment, You are looking at a drab, brownish-yellow creature, barely visible against the seafloor. Less than half a second later, that same animal is glowing with 60 pulsating rings of iridescent, almost neon blue, set against a background that has shifted to a stark, high-contrast yellow. It is one of the most dramatic visual signals in the entire animal kingdom. And the mechanism behind it is like nothing else we have found in any other cephalopod. Most cephalopods control their colors using chromatophores, tiny pigment-filled sacs embedded in the skin that can be expanded or contracted by surrounding muscles. Expand the sac, and the pigment spreads across a wider area, making the color more visible. Contract it, and the color disappears. Beneath the chromatophores, many cephalopods also have iridophores, structural reflectors made of stacked protein plates that create iridescent colors through light interference. The same principle that produces the shimmering colors on an oil slick or the surface of a soap bubble. In most squid and cuttlefish, iridophores can be switched on and off through chemical signaling, using neurotransmitters that alter the spacing or reflectance of the plates. But when researchers at Brown University bathed blue-ringed octopus skin samples in every known neurotransmitter that affects chromatophores and iridophores in other cephalopods, nothing happened. The rings did not dim. They did not change. The iridophores in the blue rings are, as scientists describe them, physiologically inert. They are always on. They are always reflecting blue-green light. So how does the octopus hide them? The answer lies in muscles and skin folds, and it is an entirely mechanical system. In the blue-ringed octopus, the iridophores that produce the blue rings are tucked inside modified skin pouches, like tiny pockets in the octopus's flesh. When the animal is calm, muscles above each ring contract and pull the overlying skin together like a pair of sliding doors, physically covering the iridophores and hiding them from view. The ring vanishes. When the octopus feels threatened, those muscles relax, and simultaneously, a ring of muscles around the outer edge of each pouch contracts, pulling the skin folds open and pushing the iridophores up closer to the surface. The ring becomes fully visible in a fraction of a second. All of these muscles are under direct neural control, meaning the signal comes straight from the brain with no chemical intermediary. This is what makes the display so blisteringly fast. The brain fires, the muscles respond, and 50 to 60 rings blaze into visibility in roughly 300 milliseconds. To amplify the effect, dark pigmented chromatophores beneath and around each ring expand at the same time, deepening the contrast between the bright blue iridescence and the surrounding skin. Critically, there are no chromatophores positioned above the rings. This is unusual among cephalopods, but it is what allows the mechanical concealment system to work without interference. The iridophores themselves are beautifully engineered. Each cell contains up to 30 densely packed reflector plates, each about 62 nanometers thick, arranged to function as a quarter-wave stack, a structure that selectively reflects light of a specific wavelength through constructive interference. In this case, the reflected wavelength peaks around 499 nanometers, which falls squarely in the blue-green portion of the visible spectrum. And because the individual plate stacks are arranged at multiple angles within the skin, the blue reflection is visible from a wide range of viewing directions, not just head-on. This matters. When a threatened octopus flashes its rings, the warning needs to be seen from every angle by every predator simultaneously. The Michelson contrast of this display, a measure of how much the bright and dark areas differ from each other, has been measured at up to 0.92 on a scale from 0 to 1, where 1 is perfect contrast. That is an extraordinarily high value. It means the blue rings are not subtle, they are screaming. And the message they carry is simple: do not touch me. What I carry inside will end you. The weapon behind that warning is Tetrodotoxin, and the story of how it gets inside the blue-ringed octopus is one of the stranger partnerships in all of biology. The octopus does not make its own venom. Not directly. Instead, it relies on colonies of bacteria living inside its posterior salivary glands, the same glands that connect to the beak, to synthesize the toxin on its behalf. Tetrodotoxin, commonly abbreviated as TTX, is a non-protein, low molecular weight neurotoxin that was first isolated and named in 1909 by Japanese scientist Yoshizumi Tahara from the ovaries of pufferfish. For decades, the toxin was associated almost exclusively with pufferfish, where it was called fugu poison, and was responsible for a grim tally of fatal poisonings in Japan every year. But as research expanded, scientists discovered TTX in an astonishing range of organisms, spanning 2 entire domains of life, bacteria and eukarya. It has been found in over 60 phylogenetically distant species of animals, including pufferfish, rough-skinned newts, poison dart frogs, certain species of crabs, starfish, flatworms, cone snails, and of course, blue-ringed octopuses. The fact that such wildly unrelated animals all carry the same molecule is one of the great puzzles of toxicology, and it points strongly toward a bacterial origin. The bacteria responsible for TTX production in blue-ringed octopuses belong to several genera, including Vibrio, Pseudomonas, Bacillus, and others. These microbes live as endosymbionts within the octopus's posterior salivary glands, which are specialized structures connected to the beak and located in the intestinal blood space. The glands have a tubulocinar exocrine structure, essentially a branching network of secretory chambers that provide a large surface area for toxin production and release. The bacteria manufacture the TTX, and the octopus stores it, deploys it through its bite, and distributes it throughout its entire body. Tetrodotoxin has been found in virtually every organ and gland of the blue-ringed octopus, including some of its most sensitive structures, the branchial hearts, the nephridia, the gills, and even the Needham's sac. This whole body distribution is significant because it means the octopus is not only venomous, able to inject toxin through a bite, but also poisonous, toxic if eaten. If a predator were to ignore the blue ring warning and eat the octopus anyway, it would ingest a potentially lethal dose of TTX from the flesh alone. Researchers demonstrated this in dramatic fashion when they documented the first known cases of wild animal fatalities, from blue-ringed octopus envenomation to adult green sea turtles each weighing approximately 110 kilograms, were found dead after accidentally swallowing tiny blue-lined octopuses, each only about 4 centimeters long. While grazing on seagrass, the octopuses had bitten the inside of the turtles' esophaguses on the way down, delivering enough TTX to fatally paralyze animals that outweighed them by a factor of several thousand. And here is where the story takes a slightly darker, more remarkable turn. The mother blue-ringed octopus does not just carry the toxin herself. She passes it to her offspring before they are even born. During egg development, the female injects tetrodotoxin and possibly the TTX-producing bacteria themselves into her eggs, priming the embryos to generate their own venom before they hatch. When the tiny hatchlings emerge, just 4 millimeters long, they are already loaded with toxins. The southern blue-ringed octopus, Hapalochlina maculosa, is considered venomous from birth, well before its signature blue rings become visible, which does not happen until roughly 6 weeks after hatching. So even in their most vulnerable, most infant state, these animals are chemically armed and dangerous. The quantity of TTX inside a single blue-ringed octopus is staggering relative to its size. A 25-gram octopus, which is barely heavier than a stack of 5 or 6 coins, carries enough venom to fatally paralyze 10 75-kilogram humans. Some estimates go even higher, suggesting that the venom load of a single animal could theoretically kill up to 26 adults. And crucially, TTX is heat stable, meaning it does not break down with cooking or boiling. There have been recorded cases of food poisoning from people who accidentally consumed blue-ringed octopuses, not realizing what they were eating. There is no way to cook the danger out, so how does the octopus itself survive? Swimming around all day saturated in a toxin that should, by all rights, be fatal to it. The answer lies in a subtle but critical adaptation of its own sodium channels. The octopus's voltage-gated sodium channels, the very proteins that TTX targets, have evolved structural differences that reduce their binding affinity for the toxin. In essence, the octopus has rewired its own cellular hardware to be resistant to its own weapon. It is a molecular arms race in miniature, played out over millions of years of coevolution between the octopus and its endosymbiotic bacteria. To understand what tetrodotoxin does to the human body, you first need to understand how a nerve signal works, because TTX targets the most fundamental step in that process. Every time your brain tells your body to do anything, move a finger, blink an eye, take a breath, it sends an electrical signal racing down a nerve fiber. That signal is called an action potential, and it propagates by a beautifully choreographed cascade of ion movements across the nerve cell membrane. It starts when tiny gated channels in the membrane open to allow sodium ions to rush into the cell. This influx of positively charged sodium ions creates a wave of electrical depolarization that travels along the nerve like a spark along a fuse. After the sodium channels fire, potassium channels open to restore the cell's resting charge, and the signal moves on to the next section of the nerve. This process repeats itself millions of times per second across the billions of nerve fibers in your body, orchestrating every movement, every heartbeat, every breath. Tetrodotoxin attacks the very first step of that process. The TTX molecule has a guanidinium group, a positively charged molecular fragment that is roughly the same size and shape as a sodium ion. It fits perfectly into the outer mouth of the voltage-gated sodium channel, like a key that enters a lock, but then gets stuck and cannot be turned. Once TTX lodges in the channel pore, sodium ions can no longer pass through. The channel is physically plugged. The gating mechanism inside the channel still works. The nerve still tries to fire, but no sodium can flow. The action potential simply does not happen. The nerve goes silent. This is not a slow, gradual process. TTX binds to the channel from the outside, and it does so with extraordinary potency. TTX-sensitive sodium channel isoforms, which are the dominant type found in skeletal muscles, peripheral nerves, and much of the central nervous system, are blocked at concentrations in the low nanomolar range. That means it takes an almost vanishingly small number of TTX molecules to shut down a channel. The toxin is so specific and so effective that it has been used for over 60 years as a standard laboratory tool for studying sodium channel function. There are 9 known isoforms of voltage-gated sodium channels in mammals, labeled NaVone.1 through NaVone.9. TTX is potent against 6 of them. The 3 that are resistant, NaVone.5, NaVone.8, and NaVone.9, are set apart by a single amino acid difference at a critical position in the channel pore. In TTX-sensitive channels, that position is occupied by an aromatic amino acid, either tyrosine or phenylalanine, which forms a strong chemical interaction with the toxin. In TTX-resistant channels, that amino acid is replaced by cysteine or serine, which does not bind TTX nearly as well. The NaVone.5 channel is the primary sodium channel in cardiac tissue, which is why the heart often keeps beating even after TTX has shut down the rest of the body. This is a critical detail. It means that a TTX-envenomed person may have a heart that is still pumping, even as every skeletal muscle in the body has gone completely limp. The progression of symptoms in a human victim follows a terrifyingly predictable sequence. Within minutes, sometimes as few as 5 to 10, the victim begins to feel numbness and tingling around the lips, tongue, and face. This spreads to the extremities. Nausea and vomiting may follow. Then comes muscle weakness, starting in the limbs and progressing centrally. The victim may experience blurred vision, dizziness, difficulty swallowing, and loss of coordination. As the paralysis deepens, it becomes a descending, flaccid paralysis, working from the face and throat down through the body until it reaches the respiratory muscles. The diaphragm, the dome-shaped muscle at the base of the lungs that drives breathing, is a skeletal muscle. It is TTX sensitive. When it stops contracting, the victim stops breathing. And through all of this, the mind can remain fully awake. The brain itself, protected behind the blood-brain barrier, may continue to function. The victim's eyes may be fixed and dilated, their pupils unresponsive, their body completely motionless, but they are aware. They can hear. They may be able to see. They know what is happening. Medical literature describes this as a state resembling locked-in syndrome, a condition where a conscious mind is trapped inside an immobile body. It is, by any reasonable assessment, one of the most disturbing effects of any natural toxin in existence. Death, when it occurs, is usually from respiratory failure. If the diaphragm stops working and no one provides artificial ventilation, the brain is starved of oxygen and cardiac arrest follows. In some cases, death can come in as little as 20 minutes. In others, the timeline stretches to several hours. But the toxin is temporary. It is metabolized and excreted by the body. And if a victim can be kept breathing, either through mouth-to-mouth resuscitation or mechanical ventilation, the paralysis will eventually wear off. Victims who survive the first 24 hours typically recover completely, with no lasting damage. The entire ordeal, from first paralysis to full recovery, may last anywhere from 15 hours to a full day. When the blue-ringed octopus is not scaring the daylights out of scientists and beachgoers, it leads a surprisingly quiet and solitary life. These animals are homebodies. They are primarily nocturnal, spending the daylight hours tucked into crevices in rocky reefs, wedged inside empty seashells, or hidden under rubble and coral fragments in shallow coastal waters. They have been found sheltering in discarded bottles, soda cans, and other human debris on the seafloor, which makes them a genuine hazard for anyone cleaning up tide pools or reaching into underwater junk without looking. Their preferred habitats span the tropical and subtropical waters of the Pacific and Indian Oceans, from the southern coast of Japan all the way down to the temperate waters of southern Australia and Tasmania, and across the Indo-West Pacific through the Philippines, Indonesia, Papua New Guinea, Vanuatu, and the Solomon Islands. They favor shallow water, typically living at depths of less than 20 meters, though some lineages have been found at depths exceeding 100 meters. They are reef animals at heart, thriving in environments that offer plenty of hiding spots, a rich supply of small prey, and the kind of complex, rubble-strewn terrain that lets a boneless animal squeeze into spaces no predator can follow. The entrance to a blue-ringed octopus den is often easy to identify if you know what to look for. Like many octopuses, they are tidy eaters but messy housekeepers. The area directly in front of the den is typically littered with the hollowed-out shells and discarded legs of crabs and shrimp, a small garbage pile that advertises the presence of a resident hunter. And a skilled hunter it is. The blue-ringed octopus feeds primarily on small crustaceans, including crabs, hermit crabs, and shrimp, along with the occasional small fish and bivalve mollusk. Its hunting strategy is classic ambush predation. The octopus sits in hiding, camouflaged against its surroundings, and waits. When a suitable target wanders within reach, the octopus pounces, seizing the prey with its arms and pulling it toward its beak. One quick bite through the exoskeleton, and the venom flows in. TTX paralyzes the prey's muscles within seconds, effectively killing it before it can struggle or escape. The octopus then eats at its leisure, using its beak to crack open shells and its arm tips to extract every scrap of edible tissue. Some species take this a step further. The southern blue-ringed octopus, Hapalochlina maculosa, has been observed using a hunting technique where it releases its venom into the water surrounding its prey, creating a toxic cloud that paralyzes the animal before the octopus even makes contact. The prey swims into the venom, becomes immobilized, and then gets collected. It is chemical warfare in miniature, and it gives the octopus the ability to incapacitate targets without ever exposing itself to the risk of a direct attack. One particularly creative variation involves the octopus pinning down its prey and forming an airtight seal around the animal with its webbed arms, then releasing venom into that enclosed space. The prey has no choice but to breathe in the toxin through its gills, and paralysis follows within seconds. It is ruthlessly efficient. There is another fascinating detail about how the blue-ringed octopus feeds that is worth mentioning, because it reveals something about the sophistication of its hunting apparatus. The octopus actually has two sets of venom glands, an anterior pair and a posterior pair. The anterior salivary glands produce a venom cocktail that is specifically tailored for subduing prey. A mix of compounds including histamine, tryptamine, octopamine, taurine, acetylcholine, and dopamine, along with tetrodotoxin. The posterior salivary glands, which are the larger and more heavily studied of the two, are the primary production site for TTX and are the glands most directly involved in both offensive and defensive envenomation. This dual gland system means the octopus has, in essence, two different chemical toolkits available to it. One optimized for killing small crustaceans, and one for deterring much larger threats, including anything that might try to eat it. The blue-ringed octopus also employs a strategy known as chemotactile social recognition, using chemical cues detected through its arm suckers to gather information about other organisms in its environment. This sensory capability influences everything from how the octopus identifies potential prey to how it interacts with members of its own species. When hunting, the octopus may use its arms to taste a potential hiding spot, detecting chemical traces left behind by crabs or shrimp, and using that information to decide whether to investigate further or move on. This is essentially what a bloodhound does when it follows a scent trail, but the octopus does it with its arms through direct chemical contact with the substrate. Every sucker on every arm is simultaneously functioning as a nose and a fingertip. Despite its fearsome venom, the blue-ringed octopus is low on the food chain relative to many ocean predators. Juvenile blue-ringed octopuses, before their venom concentrations have fully developed, are vulnerable to predation by larger fish, moray eels, and other cephalopods. Adults rely on their camouflage as a first line of defense, their aposematic ring display as a second, and their venom as an absolute last resort. These are not aggressive animals. Faced with a potential threat, the octopus's first instinct is always to flee. If it cannot flee, it will flash its rings, Only if it is physically cornered and touched will it bite. This reluctance to deploy its weapon is one of the reasons that fatal encounters with humans, while devastating when they occur, remain extraordinarily rare. The reproductive life of the blue-ringed octopus is a concentrated, tragic, one-way journey, and it is the same for every species in the genus. These animals are what biologists call semilparous. They reproduce once, and then they die. There is no second chance, no repeat performance. The entire species perpetuates itself through a single, all-or-nothing reproductive event that costs both parents their lives. The mating ritual begins when a male, typically smaller than the female, approaches and begins to caress her with a specialized arm called the hectocotylis. This arm has been modified for reproduction, with a groove running along its length that ends in a spoon-shaped tip designed to transfer packets of sperm, called spermatophores, into the female's mantle cavity. The male grabs the female, sometimes so thoroughly that he obscures her vision entirely, and inserts the hectocotylis into her oviduct repeatedly, delivering the sperm packets. This process can last for extended periods, and in at least one species, the female eventually has to physically pry the male off when she has had enough. Males are not particularly discerning about their partners. They will attempt to mate with any member of their species they encounter, regardless of sex or size. When a male mounts another male, the encounter is typically shorter, ending when the mounting octopus withdraws its hectocotylis without transferring any spermatophores. But same-sex mounting attempts are common enough that researchers have concluded these octopuses simply cannot distinguish between males and females by sight alone. Males do adjust their behavior based on the female's reproductive history. Studies on the southern blue-ringed octopus have shown that males will spend more time mating with a female who has not recently mated with another male, and they are less likely to terminate copulation early with these fresh partners. Females, for their part, can store sperm from multiple males and may use a mix of different males' sperm when they finally fertilize their eggs. Though whether they actively select which sperm to use, a process called cryptic female choice, remains an open and actively debated question in the scientific literature. After mating, the female begins the final chapter of her life. She lays a single clutch of approximately 50 eggs, though some estimates place the number as high as 100, depending on the species. In the southern blue-ringed octopus, the female does not attach her eggs to rocks or coral the way many other octopus species do. Instead, she carries them directly, cradling them underneath her arms, passing them from sucker to sucker, holding them against her body in what researchers have described as a brooding posture. She will spend the next several months in this position, rarely moving, rarely hunting, and eventually not eating at all. During the incubation period, which lasts roughly 6 months for most species, the female devotes herself entirely to the care of her eggs. She gently blows currents of water over them to keep them oxygenated and clean. She protects them from parasites, occasionally nibbling and licking the eggs to remove any growths or organisms that might threaten the developing embryos. She shields them from predators, and she starves. She has not eaten for months, Whatever fat reserves she built up during her pre-mating hunting period are all she has to sustain her through this marathon of maternal care. About a week before hatching, the embryos inside the eggs rotate, flipping so that they are positioned to emerge mantle first from the egg capsule. When they finally hatch, the baby octopuses are minuscule, just 4 millimeters long, barely visible to the human eye. In the southern blue-ringed octopus, the hatchlings are benthic from the start, meaning they settle directly onto the seafloor rather than drifting as plankton. They are venomous from birth, although their blue rings will not appear for another 6 weeks. They grow quickly, reaching sexual maturity in as little as 4 months, and the entire life cycle from hatching to death may take as little as 7 months in some species, and up to 2 years in others. And the mother, she dies. Shortly after the eggs hatch, her body gives out. A chemical secretion from the optic gland, a small organ located between her eyes that functions similarly to the pituitary gland in mammals, triggers a cascade that suppresses her appetite, accelerates her decline, and ultimately causes her death. It is programmed senescence, a biological countdown hardwired into her DNA. The father, for his part, typically dies within a few months of mating as well, sometimes sooner. Neither parent will ever see their offspring grow, given the terrifying potency of its venom. You might expect the blue-ringed octopus to be responsible for a massive toll of human fatalities. The reality is very different. Fatal encounters are exceptionally rare, and the total number of confirmed deaths attributed to blue-ringed octopus bites throughout recorded history is surprisingly low. Estimates of documented human fatalities range from as few as 3 to as many as 16, depending on the source and how the counting is done. But most toxicology scholars agree that at least 11 deaths have been recorded. In the broader context of venomous animal fatalities, this is a tiny number. More people die each year from bee stings, dog bites, or lightning strikes. The reason for this low fatality count is a combination of factors. Blue-ringed octopuses are not aggressive. They are small, secretive, nocturnal, and they spend most of their time hiding. Human encounters typically only happen when someone reaches into a tide pool, picks up a shell, or handles the octopus directly, either out of curiosity, ignorance, or the misguided belief that such a small animal cannot possibly be dangerous. And in many of those cases, the octopus bites defensively, but does not inject a full dose of venom. Not all bites result in serious envenomation, but when a full envenomation does occur, the consequences are swift and devastating, and the case reports in the medical literature are harrowing. One of the most well-documented cases involves a 4-year-old boy in Queensland, Australia, in 2006. The child was playing in rock pools at a popular beach when his mother saw him holding a small, gray-yellow octopus about the size of a golf ball. Within 10 minutes of being bitten, the boy had vomited 3 times, lost the ability to stand, and was complaining of blurred vision. By the time the ambulance delivered him to the local emergency department, 20 minutes after the bite, he had acute and progressive skeletal muscle weakness. Doctors intubated him and placed him on mechanical ventilation within 30 minutes of the bite. He was transferred to a pediatric intensive care unit, where he remained on the ventilator for a total of 17 hours. Spontaneous muscular activity began returning around the 15-hour mark. He was discharged with no long-term complications. That child survived because his mother recognized the danger quickly, and because medical help arrived fast enough to keep him breathing through the paralysis. Without that ventilator, the outcome would almost certainly have been fatal. In March 2023, a woman swimming at a beach near Sydney, Australia, picked up a small shell from the water, and a blue-ringed octopus fell out onto her stomach and bit her twice on her abdomen before she could react. She experienced abdominal pain and was treated with cold compresses before being transported to a hospital for monitoring. She escaped relatively unharmed, and the medical community was not entirely sure why. Possibly because the octopus did not inject a full venom dose, or because the bites did not achieve deep tissue penetration. Perhaps the most troubling trend in recent years is the rise of social media encounters. Multiple people have posted videos of themselves holding wild blue-ringed octopuses in their bare hands, completely unaware that they were handling one of the most venomous animals on Earth. In one widely shared incident from 2021, a woman filmed herself holding a blue-ringed octopus in Bali, not recognizing what it was. She was not bitten and suffered no harm, but toxicologists and marine biologists reacted with alarm, noting that any one of those casual, camera-ready encounters could have ended in a medical emergency. The first aid protocol for a blue-ringed octopus bite is straightforward in principle, but demanding in execution. Apply pressure to the wound. Begin artificial respiration immediately if the victim shows any signs of respiratory difficulty, and keep the person breathing until medical help arrives. There is no antivenom. There is no antitoxin in development that has been approved for human use, though a primary research report has indicated that a monoclonal antibody specific to tetrodotoxin is being explored by researchers who have shown some effectiveness in reducing toxin lethality in mouse studies. For now, treatment is entirely supportive. The goal is to maintain oxygen supply to the brain and vital organs until the body metabolizes and excretes the TTX, which typically takes 12 to 24 hours. If a victim can be kept alive through that window, they will almost always recover fully. The toxin does not cause permanent tissue damage. It does not destroy nerve cells. It simply blocks them temporarily, and then it is gone. What makes blue-ringed octopus bites particularly treacherous, beyond the venom itself, is how easy they are to miss. The bite is tiny, often producing nothing more than a small puncture that might yield a single drop of blood. Many victims report feeling no pain at all, or at most a sensation comparable to a mild bee sting. There is no dramatic wound, no swelling, no redness, nothing that screams medical emergency, until the paralysis begins to set in minutes later. Medical literature describes the signature presentation of a blue-ringed octopus bite as collapse with paralysis on or near a beach shortly after a minor bite. If bystanders do not know what to look for, they may not connect the victim's sudden weakness and confusion to the small, unassuming animal they were handling moments earlier. Every second of delay in initiating artificial respiration reduces the victim's chance of survival. It is also worth noting that the octopus's venom is heat stable, meaning that TTX does not break down when exposed to high temperatures, unlike some venoms that can be neutralized by applying heat to the bite site. This approach is completely ineffective against tetrodotoxin. Applying hot water or a heat pack to a blue-ringed octopus bite does nothing to the toxin. The only intervention that saves lives is keeping the victim breathing. So where does the blue-ringed octopus stand today? And what does its future look like? The answer, like so many things about this animal, is complicated. The International Union for Conservation of Nature currently lists the blue-ringed octopus as least concern, meaning that, based on available data, the genus is not considered to be at immediate risk of extinction. But that classification comes with a significant caveat. We do not actually know how many blue-ringed octopuses there are. Population estimates for these animals are essentially non-existent. They are small, cryptic, nocturnal, and exceptionally difficult to survey. Scientists cannot say with confidence whether their numbers are stable, declining, or increasing. The least concern label reflects a lack of alarming data more than it reflects a confirmed abundance of animals. What we do know is that the habitats these octopuses depend on are under serious and growing pressure. Blue-ringed octopuses are creatures of coral reefs, rocky seafloors, seagrass beds, and shallow tide pools, all of which are being degraded by human activity. Coral reefs worldwide are suffering from warming ocean temperatures, coral bleaching events, and ocean acidification, a process driven by rising atmospheric carbon dioxide that lowers the pH of seawater and can dissolve the calcium carbonate structures that corals and many other marine organisms depend on. Seagrass meadows, another key habitat for several blue-ringed octopus species, are in global decline, threatened by coastal development, nutrient runoff, dredging, and boat anchoring. When these habitats disappear, the octopuses that live in them lose their hiding places, their hunting grounds, and their nurseries. Additional threats include bioprospecting, the collection of marine organisms for pharmaceutical and biotechnology research, overfishing, which can disrupt the food webs that support blue-ringed octopus prey species, and the international aquarium trade, which drives the collection of these animals from the wild. Blue-ringed octopuses are undeniably beautiful, and their dramatic appearance and dangerous reputation make them appealing to exotic pet enthusiasts. But they are poorly suited to captivity. They have short lifespans, specialized dietary needs, extraordinary escape abilities, and obviously, a venom payload that makes them extremely dangerous to keep in a home aquarium. Any serious lapse in tank security could mean a lethal encounter with a curious child or an unsuspecting visitor. On a more hopeful note, the blue-ringed octopus may have something extraordinary to offer human medicine. The very toxin that makes it so dangerous is also showing genuine promise as a therapeutic compound. Tetrodotoxin's ability to selectively block specific sodium channel isoforms has attracted intense research interest in the field of pain management because certain TTX-sensitive sodium channels, particularly NaV0.3, NaV0.6, and NaV0.7, become abnormally overexpressed during chronic pain conditions like neuropathic pain and cancer-related pain. Researchers have been investigating whether carefully controlled sublethal doses of TTX can be used to quiet those overactive channels and provide relief where conventional painkillers fail. The results so far are cautiously encouraging. Preclinical studies in rodents have shown that TTX can significantly reduce neuropathic pain at doses far below those that cause systemic toxicity. The compound has progressed to clinical trials in humans, with Phase 2 and Phase 3 studies evaluating its effectiveness against chemotherapy-induced neuropathic pain and cancer-related pain, conditions that are notoriously difficult to manage with existing drugs. In one particularly creative approach, researchers at Boston Children's Hospital developed a slow-release polymer formulation that binds TTX and delivers it in a controlled trickle directly to targeted nerves, paired with a chemical penetration enhancer that makes the nerve tissue more permeable. In rat models, this system achieved a nerve block lasting up to 3 days with minimal toxicity and no tissue damage. If that technology can be safely translated to humans, it could offer a powerful, non-opioid alternative for chronic pain management, a finding with enormous implications in an era where opioid addiction and overdose deaths remain a global crisis. There is also preliminary research exploring TTX's potential to treat heroin and cocaine withdrawal symptoms, spinal cord injuries, brain trauma, and even certain types of tumors. The compound's extraordinary specificity, the fact that it targets only certain sodium channel subtypes while leaving others untouched, makes it a uniquely precise tool in a pharmaceutical landscape that desperately needs more targeted therapies. The blue-ringed octopus, an animal that most people know only as a danger to be avoided, may end up being one of the ocean's most valuable contributions to human health. And that brings us to the larger question that hangs over every creature like this. Every small, specialized, habitat-dependent species that carries secrets we have barely begun to unlock. What happens when we lose the habitat before we finish the research? What cures? What treatments? What breakthroughs go extinct before we even know to look for them? The ocean is full of organisms whose biochemistry we have barely scratched the surface of. And the blue-ringed octopus is a perfect example of why protecting marine biodiversity is not just an ecological imperative, but a practical, medical, and scientific one. Every reef that bleaches, every tide pool that gets paved over for a resort or a marina, every seagrass meadow that dies back under nutrient pollution represents a potential loss not just of individual animals, but of the evolutionary innovations those animals carry. Innovations that took millions of years to develop, and that we may never be able to replicate synthetically. The blue-ringed octopus has been perfecting its chemistry for 30 million years. It evolved a venom delivery system that is the envy of pharmaceutical engineers. It invented a mechanical light display that inspired engineers at the University of California, Irvine, to develop new approaches to adaptive camouflage and infrared signaling technology. It lives a life so compressed and so ferociously efficient that it is born venomous, reaches maturity in months, mates once, and dies before it is 2 years old. And yet, in that brief span, it plays a critical role in controlling crustacean populations and maintaining the balance of reef ecosystems. It is also one of the only cephalopods known to use chemotactile recognition to influence its social and predatory behavior, a trait that researchers believe has been a driving force behind many of its evolved behavioral patterns. From the way it selects prey to the way it chooses mates, it is soft, it is small, it is shy, and it is sitting right now in a tide pool somewhere off the coast of Australia, tucked inside a discarded soda can, perfectly camouflaged, waiting for a shrimp to wander past. If you disturbed it, it would flash 60 blue rings in a third of a second. A last, beautiful warning from an animal that carries more chemical firepower per gram of body weight than almost any other creature on Earth. Leave it alone. Let it hunt. Let it live out its impossibly brief, impossibly intense life. And maybe, if we protect the reefs and tide pools it calls home, it will keep teaching us things we never knew we needed to learn. The blue-ringed octopus is proof that the most extraordinary things in nature often come in the smallest, quietest packages. And sometimes, the tiniest body holds the biggest lessons about what it means to survive.